Chapter 28, this is our reproductive system lecture. So we're going to start talking about the common elements between the two systems. Both females and males have primary reproductive organs called the gonads. The ovaries in females, the testes in males, they're homologous structures. They produce sex cells called gametes that unite at fertilization to initiate the formation of a new individual. When they come, they both produce large amounts of sex hormones that affect maturation, development, and activities of reproductive organs. There are also accessory reproductive organs, such as ducts, to carry the gametes away towards fertilization or outside of the body. The organs are used for sexual union, copulation, coitus, sexual intercourse. If fertilization occurs, the female reproductive tract provides support, protection, and nourishment. Part of evolutionary theory for just kind of why we have developed, even in like modern societies, why we still have very kind of traditional roles for females, is because in smaller mammals and smaller animals, the energy that it takes to produce an egg and then to care for that young, to feed the young and care for it and let it and help it to grow, is so much that it really does occupy in smaller animals a lot of energy from a female, and so there's kind of been this translation to even in higher mammals where clearly a woman can both care for a child and also be a fully functioning member of society. We still kind of have these same kind of stereotypes about women and the energy that it takes to be able to do something as simple as, not, I wouldn't say simple, but do something as common as ovulate. There is, we don't really need to kind of keep those, but we do still. So anyway, we'll talk about that more as this lecture goes on. But essentially that's what the female tract is going to be able to do. During puberty, you have the onset of adolescence, external sex characteristics develop, your secondary sexual characteristics. The gametes also begin to mature, the sex cells inside of the gonads. The gonads will also start to secrete sex hormones. We'll look at hormonal regulation of both males and females fairly in depth, but you should also just understanding it overall, there are some common elements to it. The hypothalamus starts secreting gonadotropin-releasing hormone. You should know both the name and the abbreviations for all of these. It will stimulate the anterior pituitary to release two different hormones, both FSH and LH, your follicle-stimulating hormone and your luteinizing hormone. These will signal for the gonads to start producing significant levels of sex hormones and start the process of gamete maturation. Both of these in males and females. So the gametes, they're in both sexes. The female produces and releases a single gamete monthly, the oocyte. That's typical. Sometimes it's more, sometimes it's less, but we always kind of deal with just the base average in classes, especially introductory classes. So typically you have one oocyte released a month. The male produces large numbers of gametes, 100 million a day, stored for a short time only, and if not expelled from the body, they're resorbed. So again, kind of going back to this idea about the energy that gets put into the gametes. A female is producing, on average, typically one gamete a month for our species. Males are producing a ton more. So you can just see the energy that it goes into each of these. It takes an entire month for a single female oocyte to mature, and that's obviously so much more energy input into it. So it's, again, in smaller animals, it does require a significant amount of energy. In us, we can manage it. Females can manage it. I won't let myself in there. Okay, the perineum, it's the area between the thighs, from the pubic symphysis to the ischial tuberosities, back to your coccyx. So it's, and that always lovely fact that it may be partially torn during childbirth. They do episiotomies to try to relieve that, or to do it rather in a non-tearing way. So it's divided into two triangular bases, your anterior and posterior triangles. This is kind of structural. I'm not going to get super into this. Like, really just understand that this is your perineum. And we're good. Okay, gametogenesis. It's the process of forming human sex cells. Again, those sex cells are going to be your gametes, your eggs, and your sperm. They begin with cell division, meiosis, as opposed to mitosis, which is just going to be for your typical somatic body cells. Similar process in both males and females for the development of the gametes, but with some distinct differences. Uh, the heredity information, our DNA is carried on these gametes. On 23, and in all of our body cells have our genetic material. 23 pairs of chromosomes in the human body cells, 22 pairs of autosomes, and 1 pair of sex chromosomes. The autosomes contain genes that code for your cellular functions, determine most of your characteristics, um, eye and hair color, height, skin pigmentation, musculature, they code for your immune system, everything. All those kind of things are coded on your autosomal cells, or your autosomal chromosomes. And then together they are homologous chromosomes. So when you have a pair of matching autosomes, they're homologous chromosomes. That's your 1 through 22. The 23rd pair of chromosomes are your sex chromosomes. You have 2X or 1X and 1Y. Females are double X, males are XY. There are variations. I'm not like super into like I don't study genetic hereditary diseases that often. So I'm not going to get super. I'm not going to ask you about that at all. But just understanding that you can have XXY females, that is possible. They're usually infertile. One member of the chromosome pair inherited from each parent. So you get an X from your mother always, and then either an X or a Y from your father, and that will determine male or female. You have both diploid and haploid cells. Your autosomal body cells are going to be diploid. They have 23 pairs of chromosomes. We represent that as 2N, meaning N is the unpaired number of chromosomes. So a diploid cell is a 2N cell. It has 23 pairs of chromosomes, 46 individual ones. A haploid cell is just N. Its haploid number is N. It has 23 chromosomes, not 23 pairs. These are going to be your gametes. This makes sure that the offspring don't end up with 4N total chromosomes. So your gametes, your egg and your sperm, are each only going to have half of your genetic information. And we'll see how that happens, because essentially as they start as one cell, they go through a meiotic division, which ends up with 4 daughter cells. And each of those daughter cells is going to have a different combination of your DNA. Or of DNA, rather. Okay, so again, you start with a diploid parent cell, so it has a 2N, 23 pairs of chromosomes. It produces haploid daughter cells that are your gametes. Begins with a diploid parent cell in the gonad, either the ovary or the testes. 23 chromosomes from the organism's mother, 23 from the father, and must undergo meiosis to produce haploid gametes. Everything I was just saying. Okay, so mitosis, it is somatic cell divisions. You've learned this produces 2 daughter cells. Excuse me. Genetically identical to the parent. Diploid daughter cells, no crossing over. So you start with a diploid, you end with a diploid. Starting with 1 parent cell, producing 2 daughter cells, genetically identical. That's why your skin typically always looks the same. And when you get a new mole or a new spot, it's always important to watch it, because that could potentially be not just a freckle, but it could be some type of growth that could be developing. And it being different is significant, because it's not supposed to be. So for meiosis, this is for sex cell division. 4 daughter cells genetically different from the parent cell. This gives you that genetic variation that allows for adaptations in individuals. You end up with haploid daughter cells. It includes crossing over, which is when genetic material is exchanged between homologous chromosomes. So remember, homologous chromosomes are essentially the same, they code for the same things. You get genes from both parents on 1 chromosome. Okay, going through the phases. Again, meiosis 1, it's going to start off just like mitosis. You have prophase, metaphase, anaphase, telophase, and cytokinesis when they start to split. And then you have a second round of division. So it starts to go through prophase 2, metaphase 2, anaphase 2, and then your final cell division. It is, why did I miss it? I'm trying to remember, because it should be that in metaphase 2 is when crossing over occurs, but okay, I'm sorry, prophase 1. That it is. Yeah, crossing over starts to occur in prophase 1. So you have these replicated chromosomes, and they essentially just switch little parts. So they just start to kind of, you can see they just kind of have exchange. You have like a red chromosome that has a little blue leg right there, and same thing, this one has a little red leg. It's because they're crossing portions of their genetic material to give even more variety. That causes, sometimes, it causes no real discernible changes because of the way genetic material is, DNA is translated and or transcribed and translated. But sometimes it does cause mutations. Most mutations, we'll get into this in more depth, but most mutations are fatal, but sometimes they can be beneficial. And so this kind of crossing over gives more genetic variety. All right, so the female gonads, the ovaries. The ovarian follicles are the site of oocyte production and sex hormone release. Your ovaries are paired oval organs within the pelvic cavity lateral to the uterus, slightly larger than an almond in the adult, varying due to menstrual cycle and pregnancy. So you have your uterus deep to your vagina. There, off to the sides, are the ovaries with the fallopian tubes, the uterine tubes, allowing for transport of the oocyte. This is a sagittal section of the ovary. You can see developing oocyte within the follicle. All right, so ovarian follicles. It's important to understand that in the ovary, you have a follicle that contains an oocyte. The follicle is there to support the ovary, to support the actual oocyte. So here is your ovary, here is a follicle, and there inside the follicle is the oocyte. And then you have your follicular cells and your antral fluid that are actually supporting the developing oocyte. So oocyte, that is your egg. That is going to be the female gamete. The ovary is the female gonad. And the follicle cell is inside of the ovary containing the oocyte. Thousands are in the cortex, 6 main types representing different stages of development, and you get to learn them all. Okay, you have your, starting with the primordial, down here, your primordial follicles. We go primary, secondary, antral, mature, that's what gets ovulated, and then the corpus luteum and the corpus albicans. Corpus luteum, corpus albicans, sorry. All right, starting with your primordial, it's the most primitive type of ovarian follicle. It contains a primary oocyte and a single flattened layer of follicular cells. This primary oocyte is arrested in the first meiotic prophase. Remember, mitosis, you have 2 stages. It's going to be in the first meiotic prophase. 1.5 million present at birth. So for all of these, you should know like the name of the follicle, kind of what stage it is relative to the other follicles, what type of oocyte it has, and with that oocyte, you should know, okay, where is that oocyte in relation to its development. From a primordial, you have a primary follicle. From maturing primordial ones, it still has a primary oocyte with a single layer of cuboidal follicular cells, now called granulosa cells. That's important. The primary follicle secretes estrogen, which will help to stimulate changes to the uterine lining that help to prepare it for implantation. So the uterus is what's going to be supporting the egg cell, the fertilized egg. So essentially, if the egg is fertilized, it implants in the uterus, and you don't want the uterus to have to just kind of start from zero and then start trying to develop. You want the uterus to be ready and ripe. If you've ever made a garden, you're not just going to buy plants the same day that you're trying to prep your garden. You're going to spend some time really preparing it, laying the groundwork, so that way then when you go to plant the flower, this is a beautiful metaphor. When you go to plant the flowers, the soil is ready and ripe for implantation. Same thing here. You have estrogen being secreted by the primary follicle. So it's not only supporting the oocyte in its development, it's also caring for it for its future implantation. Immediately surrounding the primary oocyte is the zona pellucida. Important. It's a translucent structure containing glycoproteins. So primary oocyte surrounded by these granulosa cells. Together they make the primary follicle. You have the zona pellucida, which is just kind of this area, again, of glycoproteins surrounding the oocyte, and then these other developing primordial follicles. A secondary follicle forms after that. It's still a primary oocyte. Now it has more layers of granulosa cells as well as thecal cells on the periphery. These are helping to control follicular development, secreting androgens, converted to estrogen by those granulosa cells. So again, you can see this thicker layer of the granulosa cells. The zona pellucida is also starting to thicken, and then you have these thecal cells on the outside trying to regulate that development. After that, you have the antral follicle. It's called that because it has this fluid called antrum. In that, there is serous fluid that starts to increase as as it gets closer to ovulation. This will start to force the oocyte to one side of the follicle. It starts to get surrounded by a cluster of cells called the cumuloorifice. I'll never ask you that. Immediately surrounding the oocyte is the zona pellucida. The corona radiata has now developed. That's external to the zona pellucida, an innermost layer of cumulus oorophoros cells. Again, you don't need to know necessarily that it's this specific cells, but you should know both the corona radiata and the zona pellucida. Again, you can see this antral fluid starting to develop and how the oocyte is being pushed towards one side of the cell. Lastly, you have your mature follicle that contains a secondary oocyte now. So this is kind of the first side that we've seen of that oocyte developing. It's surrounded by the zona pellucida and the corona radiata, lots of granulosa cells. It's got that fluid-filled antrum. It has gone through all of meiosis I. That's why it's now a secondary oocyte. But it's still arrested in the second meiotic metaphase. One of these is formed per month, usually. After the mature follicle, you have the corpus luteum, which is remnants of that. It's after the oocyte has been expelled, so after the follicle has expelled the egg, I'm not saying regresses, it changes into the corpus luteum. This will start to secrete progesterone and estrogens at pretty large levels, stimulating the buildup of the uterine lining, preparing the uterus for possible implantation of the fertilized oocyte. So after the oocyte has been expelled, after the oocyte has been ovulated, the corpus luteum will then start to secrete even more progesterone and estrogen to help with that uterine lining, essentially. Just kind of taking over for the first couple of weeks. If there is fertilization of the oocyte, it'll get implanted, and it will take a couple of weeks for that implantation to start before it starts secreting its own hormones. So the corpus luteum essentially just kind of takes over for those first couple of weeks after fertilization. Different stuff if fertilization doesn't happen. The albicans is formed from the regressed corpus luteum, so that's just like one of those distinctions. The corpus luteum is a transformation from a mature follicle to a corpus luteum. The corpus luteum to the corpus albicans is a regression. You have connective tissue that starts to replace your functional tissue, and most structures are completely restored. It stops secreting any kind of hormones. Okay, so oogenesis, that was the development of the follicle that is supporting the oocyte. Now let's look at the actual progression of oocyte development. Going from a primary to a secondary oocyte, occurring over the stage of the life of a female. Remember, it's only that one egg that is ovulated each month. That's the one that develops into a secondary oocyte. The rest of them are paused in a primary oocyte stage. So before birth, during uterine development, the ovary contains primordial germ cells called oogonia. These are diploid cells. They go through mitosis to produce primary oocytes. They start meiosis, but they stop in prophase I, and that's when they become primary oocytes. Females have 1.5 million within the ovaries, and they stay in prophase I until after puberty. Let's see if I want to say anything else about this. No. Okay. So during childhood, before puberty, the ovaries are inactive. You have atresia that occurs, which is regression of some of those primordial follicles. So by puberty, about 400,000 follicles remain in the ovaries. And again, those are going to contain your oocytes. So about 400,000 eggs present at puberty, roughly. So from puberty to menopause, the hypothalamus is going to release those gonadotropin-releasing hormone. That will stimulate follicle-stimulating hormone and luteinizing hormone. So now that you've kind of learned about follicle development, you can notice follicle-stimulating hormone. That should give you some idea about what it's going to be doing. These levels vary in cyclical patterns, produce a monthly sequence of events called your ovarian cycle, which has 3 phases: follicular, ovulation, and luteal. So let's just look at our hormones: follicle-stimulating luteinizing hormone, follicular phase, luteal phase. We can make some connections. I always like to go ahead and just step in and say, produce monthly sequence of events. Sometimes it's longer, sometimes it's shorter. It's not always a perfect. We'll see. We work with a 28-day cycle. It's not always a perfect 28-day cycle. This is probably not news for most of the females in the class, but for some of the males, it's important to understand that you can learn. They do occur in these stages. That's not going to change, but the actual length and duration is going to vary person to person. So the first phase, this follicular phase, it's days 1 to 13. We'll say like the first phase of the cycle. You have around 20 primordial follicles stimulated to mature into primary follicles by your LH and FSH. So 20 primordial follicles out of the, let's say that this might be the first menarche, so like the first time that a female is going through puberty. So you start with 400,000 follicles. 20, just randomly 20 of those are going to start to develop into primary follicles. No idea why. It's just 20 random ones get picked. The granulosa cells, as they develop, will release inhibin to prevent further FSH production. So you don't have excessive follicle development. That way you kind of end up with roughly 20. And we'll see that, obviously a female isn't releasing 20 eggs. So we'll see that that's kind of, the magic number-ish, that if you start with 20 primordial follicles developed into primary ones, by the time you get to the mature ones, you're only going to have one that matures fully to a mature follicle. And part of that is because the granulosa cells of those follicles are releasing this inhibin hormone, inhibiting FSH, which causes negative feedback. Few primary follicles mature into secondary. Usually only one matures to the vesicular follicle. The volume of fluid increases within the follicular antrum, again forcing the oocyte to one side. The primary oocyte in the antral follicle finishes meiosis 1 and forms 2 cells. It's going to be one polar body, which is non-functional, receiving minimal cytoplasm, and regresses. So essentially you have that oocyte, that egg cell, that's going to be going from a primary to a secondary oocyte. It's going to finish meiosis 1, and instead of having equal kind of cell sizes, you're going to have one that's just like a kind of puny little nothing really, that literally just gets broken back down, and you're going to have one that is derived from the primary oocyte that receives the bulk of cytoplasm. It develops fully into the secondary oocyte. It reaches metaphase 2 before pausing again, and completes meiosis if it's fertilized. If not, it breaks down in 24 hours. So after ovulation, if it is not fertilized within 24 hours, it'll start to break down. So that was your follicular phase. Your ovulation is the release of secondary oocyte from the mature follicle. It occurs on day 14 of the 28-day cycle. Again, women can track their cycles and can use that as a marker. Usually only one ovary ovulates each month, induced with a peak in LH secretion. The antrum increases in size and swelling. That fluid starts to increase, expands until the ovarian surface starts to thin, so it actually will push up against the ovary so that the ovary starts to thin out, and eventually ruptures, expelling your secondary oocyte. Again, these are microscopic, like tip of a pin. So when it talks about like rupturing the thin surface, by the fact that it's thin, it makes it easier just to reveal. After that, you have your luteal phase. Remember your corpus luteum. The remaining follicle cells becomes the corpus luteum. It's the end of the cycle. The corpus luteum is a temporary endocrine gland, meaning that it's going to be secreting hormones, progesterone and estrogen, to build up the uterine lining, preparing for a fertilized oocyte. Lifespan is about 2 weeks if the oocyte is not fertilized. The corpus luteum starts to regress, causing a drop in progesterone and estrogen, which again, if there is no fertilization, will cause the shedding of the uterine lining, which we know as menstruation. Shed lining is specifically called the menses, and it marks the end of the luteal phase. Your first menstrual cycle is menarche, usually around age 11 or 12. Varies quite a lot. Here is the process you should understand this whole chart, both the follicular development over on this side, as well as the process of oogenesis, before birth, childhood, and puberty. I'm trying to see, yeah, all of this is good information. You should know the stage of, that starts by, like, this is uterine development, so it goes through first a mitotic division, and then meiotic divisions. You should know which phase of meiosis it's pausing in. You should understand what the polar body is and what's happening to that, as well as how it pairs with your follicle. So this is another chart that I love getting to use because it just summarizes so much of the information for us. You have your FSH and LH levels. You can follow the different phases of follicular, ovulation, and luteal phases. You can track your uterus lining. We'll get into these next 3 phases soon, as well as the hormone levels. You can see that when you have this peak in estrogen, you have this peak in your LH, ovulation. After ovulation, once you get into your luteal phase, you can track your luteal phase with peaks in both progesterone and another little one here of estrogen, and then compare that to the uterine lining. All right, menopause. Women see cycling for 1 year. That is called menopause. So after not having a menstrual cycle for 1 year, it is officially menopause. Assuming that you are not pregnant or any other medical conditions. Normal onset is ages 45 to 55, varies. No more ovarian follicles remain or the follicle maturation stops. Significant amounts of progesterone and estrogen are no longer secreted. The uterine lining no longer growing, so no further menstrual periods. All right, now let's get into the ovarian cycle in depth. Y'all thought we were done with it. Now we're going to look at it even more. All right, so your hypothalamus begins this by secreting gonadotropin releasing hormone. That is going to trigger the anterior pituitary to release FSH and LH. Those 2 hormones are going to target the ovaries, stimulate follicular development, causing the follicles to mature and affecting secretion of other hormones, right? Maturing ovarian follicles will secrete both inhibin and estrogen. The inhibin is going to, both will have a negative effect on the hypothalamus and the anterior pituitary, so try to limit the FSH-LH release. Estrogen assisting the development of the mature ovarian follicle, and that mature follicle producing a larger amount of estrogen, initiating a positive feedback loop to help develop itself. That feedback loop results in an LH surge from the anterior pituitary. Without that surge, you have no ovulation. The corpus luteum forms from the ovulated follicle. The luteum secretes progesterone, estrogen, and inhibin, inhibits the hypothalamus and anterior pituitary, and builds up the uterine lining with estrogen and progesterone. Again, degenerates after 2 weeks if it's not fertilized, if no fertilization occurs in the oocyte. And the cycle repeats. LH levels are reduced by negative feedback events, estrogen, progesterone, and inhibin drop, and as those levels drop, the hypothalamus can then start to secrete GnRH again, and the cycle starts over once more. If the secondary oocyte is fertilized and implants, it is called a pre-embryo, and it will start to secrete human chorionic gonadotropin. That is what is detected on pregnancy tests. This will mimic the effects of LH and continues to stimulate that corpus luteum so that it doesn't regress. That will then keep producing estrogen and progesterone to help maintain the uterine lining until the placenta can take over and produce its own progesterone and estrogen after 3 months. By that point, the corpus luteum will then again regress into the corpus albicans. So if you do have fertilization, it gets implanted, it starts secreting this HCG. That's going to then support, because you'll have a drop in luteinizing hormone, and so HCG will stimulate and support the corpus luteum. That will then continue to provide estrogen and progesterone to support the uterine lining as the embryo is embedding. After about the first trimester, the corpus luteum will fully regress and the placenta is fully developed to be able to support the developing embryo. Here you can just look at the follicular phase, late follicular, ovulation, and luteal phase hormones, being able to understand what is both inhibiting and what is causing positive feedback. All right, ovarian cancer, primary malignancy in the ovary, may originate from oocytes, connective tissue, or surface epithelium. It is the fifth most common cancer in women. Non-specific symptoms: constipation, urinary urgency, abdominal swelling, pelvic pain, indigestion, nausea, back pain, weight changes. So really nothing, again, very random symptoms. No reliable tests. It's often detected late, unfortunately. All right, connecting the ovaries to the uterus, you have your uterine tubes extending laterally, transporting the oocytes, covered and suspended by mesosalpinx. Specific superior part of the broad ligament. Remember, the broad ligament kind of covers that entire the uterus and ovaries. The first part of the uterine tube is the infundibulum, and when I say first part, I mean the part closest to the ovary. It has those fimbriae that extend and kind of wrap around the ovary, that enclose the ovary at the time of ovulation. Otherwise, they are drawn back. Next is the ampulla. This is where fertilization typically occurs. Then you have the isthmus, which is the longer part, and the uterine tube, which penetrates the wall of the uterus. I've done this in the lab, so I'm not going to spend a lot of time. The walls of the uterine tube, there's the mucosa, muscularis, and serosa layers. The mucosa, simple ciliated, linear fold to reduce the size. It has cilia on the apical surface, so closest to the lumen, to beat in the direction of the uterus and drawing the oocyte in towards the uterus. Muscularis layer is just what you think, intercircular, outer longitudinal, with contractions to help propel the proembryo through the uterine tube if it's fertilized. And then in serosa, an external serous membrane. You can have an ectopic pregnancy. Fertilized oocyte implants outside of the endometrium, so outside of the uterus. It could be usually along the tube. One type of ectopic pregnancy, fertilized egg implants in the uterine tube. It is the uterine tube is unable to expand as the embryo grows. The embryo is only viable until week 8. The uterine wall will rupture if the embryo isn't removed, and hemorrhage and loss of life is possible. There are a lot of debates still going on about the appropriateness of abortions, whether they are elected or for life-saving procedures. But this is one of those situations where it's very clear that if something does not happen, the embryo isn't viable at this point. Once it has started to implant into the uterine tube, there's really nothing that can happen, and not removing it would only cause further loss of life for the mother. So, regardless of personal opinions, there are some things that science has to look at. All right, your uterus then is the pear-shaped muscular organ. The lumen is on the inside of it. It connects to the uterine tubes and the vagina. It is the implantation site for a pre-embryo. Notice that we don't call it a baby. We're not even calling it a fetus at this point. It is a pre-embryo. After it implants, it becomes an embryo, which then becomes a fetus, and then after birth, it becomes a baby, child, infant, lots of other words for it. But for our references, that's what we're talking about. And for your answers in your tests, please use those appropriate terms. The uterus is going to support, protect, and nourish a developing embryo. It forms a vascular connection that develops into the placenta. It will eventually eject the fetus. It contracts and sheds the lining if the oocyte is not fertilized, and receives blood from uterine arteries, which are branches of your internal iliac artery. If you remember, that comes off of your abdominal aorta. The different regions, you have the fundus body, the isthmus, and the cervix. The body is the major portion, the isthmus is the narrow constricted inferior region, and the cervix projects into the vagina and kind of is a small canal between the uterus and the vagina. Or the lumen of the uterus, I should say. There are 3 walls to the 3 tunics to the uterus. Your perimetrium is your outer tunic, continuous again with that broad ligament that's just connecting all of those pelvic structures. The middle thick layer is the myometrium, your muscle layer. It has, again, 3 intertwining layers of smooth muscle, as opposed to just normal 2. And then the endometrium is the innermost mucosal tunic. It has simple columnar epithelium. Your deeper basal layer is your permanent layer. It undergoes few changes during each uterine cycle, but the more superficial functional layer begins at puberty, grows from the basal layer under estrogen and progesterone, and if no fertilization, it is shed. So this is the one that these hormones are going to be stimulating and helping to grow. The vagina is a thick-walled tube. It connects the uterus with outside of the body, functions solely as a birth canal. It is, in males, we'll see that the reproductive structure has a urogenital function as well. In females, the reproductive tube only is a reproductive structure. Passes away from menstruation, receives the penis during intercourse. Intercourse for copulation. The vagina is supplied by vaginal arteries, drained by vaginal veins. Thin, distensible wall should be able to stretch and accommodate both the penis and the expulsion of a fetus. Your mucosa is non-keratinized, stratified squamous epithelium, so that should make sense. It can take some abrasive structures, or can take some abrasion. Highly vascularized, acidic secretions to help prevent infections. Inferior region with numerous transverse folds projecting to the lumen near the vaginal opening, which is your vaginal orifice. Form a vascularized membranous layer called the hymen. It is not appropriate in all of the teaching that I have learned to use the hymen as some type of, we can get a whole discussion about the cultural impacts of the concept of virginity and how virginity is really a cultural concept and not so much an actual, we discuss it in medicine. A lot of times the hymen is used as a indicator of virginity. That's just straight up how it's been used. But there are a lot of reasons why a hymen might be torn. It could be trauma. It could just not have been fully formed. It could be lots of different things can cause for a hymen to be torn. And again, just the concept of virginity is a cultural one. I can keep on talking about that. This is always one of those lectures where I have to think ahead before I say things because sometimes I talk too much. All right, the muscularis layer has the outer and inner layers of the smooth muscle. And the adventitia contains inner elastic fibers and connective tissue. There you go. Endometriosis is when part of the endometrium, that inner layer of the uterus, is displaced. It becomes implanted on external surfaces of organs. It can grow under the influence of some hormones. It can't be expelled normally. It can cause awful, awful pain and scarring. And it can be linked to fertility issues. There are hormones to treat the growth of endometrial tissue or surgery. From what I have heard most of the time, it's birth control that people are offered, which seems to be a common solution for a lot of female issues. Whether or not that's the most effective treatment, there is still ongoing research. Cervical cancer, common malignancy of the female reproductive system. Most important risk factor is the HPV infection, the human papillomavirus. There are two forms of it. One of them is essentially benign. Benign isn't the right word, but I can't think of it right now. It is not actively causing any type of infection. It is essentially harmless. You just become a carrier for it. And the other kind can be very aggressive and cause cervical cancer. A vaccine is available for the four most common types. You do need to receive it fairly. I believe they start giving it around 12 or 13 years old. Pap smear detects cervical cancer in early stage. Epithelial cells are scraped from the cervix edge and examined for abnormal development. You should be getting checked regularly. If cancer cells are present, a portion of the cervix is removed. If it is fully invasive, the entire uterus can be removed, called a hysterectomy. We really need to change that term. That is not a good word for us to keep using. Okay, the uterine cycle. So the uterine cycle are the cyclical changes in the endometrial lining. So we did our follicle and our egg development. Now let's look at our uterine cycle and uterus development. It is regulated by estrogen and progesterone and has three distinct phases: the menstrual, proliferative, and secretory phase. Again, it's based on that 28-day cycle. But again, it varies, here at least they finally mentioned, varies 21 to 35 days. So variety for sure. The menstrual is the first part. It's that sloughing off of the functional layer, and it lasts through menstrual bleeding. So this is kind of those first few days. It will vary. Some women will have less flow, some women will be more. It just depends. The proliferative phase is the development of that new functional layer. So that's, again, what was sloughed off in the menstrual layer. It overlaps time of follicle growth and ovary estrogen secretion. And then the secretory phase, increased progesterone from the corpus luteum results in increased vascularization and uterine gland development. If fertilization does not occur, the corpus luteum will degenerate. You have a big drop in progesterone, and without that, the functional layer will slough off. Once it sloughs off, you start again in your menstrual phase. So again, starting with this menstrual phase, your functional layer drops off. You have this starting to peak as your follicle and oocytes start to develop. They start to secrete more estrogen. That will start to build back up this functional layer. Then you have ovulation, and the corpus luteum starts to secrete progesterone, which really then kicks up this uterine development before it then starts to slough off again if no fertilization occurs. Remember, if fertilization does occur, you then have the pre-embryo secreting HCG, which keeps the corpus luteum supported, so that way it can continue to secrete progesterone, providing growth for the uterine lining. Okay, now mammary glands. Each located within the anterior thoracic wall, composed of exocrine glands, secrete breast milk, which contains proteins, fats, lactose for infant nutrition. You have both a nipple and an areola. Nipple is the center projection of the breast, multiple tiny openings of excretory ducts transporting breast milk. The areola is the pigmented ring around the nipple, uneven surface due to sebaceous glands. It is often darker in women after they have given birth, called parous. Once you have been given birth, you're parous. Lighter in women who hasn't, called nulliparous. You can divide the glands into lobes, further divided into lobules with alveoli, producing milk in a lactating female. They become more numerous and larger in pregnancy. They're drained by lactiferous ducts, expand to form sinuses where the milk becomes stored. Fairly straightforward. Lactation is the actual release of that breast milk occurring in response to internal and external stimuli. It's true. If a baby cries, there is a chance that a woman will start lactating. It's a hormonal thing. They start to produce after giving birth. Prolactin is produced in the anterior pituitary, responsible for milk production. So prolactin produces milk. With the increase, the mammary gland forms more and larger alveoli as prolactin levels increase. Your oxytocin is responsible for milk ejection. So we say that prolactin makes the milk. Oxytocin allows it for letdown, is what it's called. When milk is released or ejected, it's the milk letdown, and that's from oxytocin. It's produced by the hypothalamus, stored in the posterior pituitary. Remember, the posterior pituitary has ADH and oxytocin. Breast cancer occurs in 1 in 8 women in the US. Several risk factors: family history, longer reproductive span, obesity, nulliparity, late age, specific genes, the BRCA1 and 2. Most related to increased estrogen exposure arise from the duct epithelium. You can have mammograms to detect small malignancies early on. Should be done every 1 to 2 years in women over 40. Okay, that was all the women stuff. So I always like to take this moment. A lot of times, especially in classic anatomy and physiology, when I say classic, I mean when the field was developing, over the past hundreds of years, it was always studied as males were the standard because they didn't have all the pesky hormones and all the stuff that women have. And the idea is that all of these hormones that are so integral to development of the female, and as we've seen, there are so many different parts that do need to be kind of working together in female development. For classical anatomists who were all men studying this, they saw all of that stuff with the women were just extra hormones, and men were the base model, and women just were men with pesky hormones. That's how they treated it for centuries. So it's always funny to me, we kind of do this lecture, and it's like we get through all that, and then we get to the males, and it's like, okay, there's sperm, testes, and a penis, and it's pretty much it. And it really, luckily, kind of is. But I always think it's just one of those moments to like, okay, now that we got all those pesky hormones from women out of the way, let's get to the simple one, which is the men. And all jokes, definitely. Well, no, that was all actually very serious, but we can laugh at it now a little bit. All right, males have the primary reproductive organs are the testes. They're homologous to the ovaries. You have accessory organs, ducts and tubes, accessory glands, and the penis. The scrotum is the skin-covered sac that holds the testicles. It provides a cooler environment because sperm need a cooler, lower than body temperature development for development and maturation. The scrotum is homologous to the labia majora. There is a seam along the middle called the raphe. The dartos muscle is internal to the fascia of the scrotal wall. The spermatic cord has blood vessels and nerves supplying each testes, originate in the inguinal canal, has 3 layers. The fascia, it has the cremaster muscle. This is going to be from the internal oblique. Let's see, do we talk about it here? I think we do soon. You have a testicular artery from the abdominal aorta within that spermatic cord as well. The pampiniform plexus, remember a plexus is just a network of veins. It's a network of veins surrounding the testicular artery. It pre-cools arterial blood prior to reaching those testes. I think I said, did I say network of nerves? I hope I didn't. Plexus is just a network of any kind of tubular system. So the scrotum will respond to temperature changes. This is what I was looking for. With elevated temperature, you have a relaxation of the dartos muscle to lower the testicles away from the body to help cool them. The cremaster muscle relaxes to allow the testes to move down as well. And then with decreased temperature outside, you have a contraction of both of those muscles to pull them in closer to the body to help conserve heat. You can see the testicles again out here outside of the body. They can reside back up into the body. And sometimes if you do have quite a shock, like a big shock into cold, they can disappear. They come back down eventually. Scrotum testicles. One thing you can see slightly in this one, but definitely more in this image, there's always one that's going to hang lower than the other. That's just a nature of testicles. Okay, so more about them. They are relatively small organs housed within the scrotum, producing sperm and androgens. Covered anteriorly by a serous membrane, your tunica vaginalis. It's part of the abdominal peritoneum. Has a parietal and visceral layer with a cavity with serous fluid. Not going to get like too in depth into this, but again, just kind of recognizing, okay, how many times have we seen a cavity that has a parietal and a visceral layer with serous fluid in between? This has been in the testicles. We've seen it in the abdominal pelvic cavity. Within your thoracic cavity, inside of your heart, like this is a common structure, right? This is a common motif that's repeated over and over. Inside the testes are these seminiferous tubules, very convoluted and elongated. That helps them to fit more into one structure. You have up to 4 per lobule. They have non-dividing support cells called your sustentacular cells to nourish the developing sperm. They release inhibin when the sperm count gets too high. That inhibits FSH. The seminiferous tubules contain your dividing germ cells continuously producing sperm. Remember, a germ cell is always a cell that it will stay the same and produce progeny. So you will produce, let's say it splits through a mitotic division. You have one germ cell that splits into 2. One of those will remain a germ cell, and that second one will go on to continue. That germ cell will continue dividing. So technically your germ cell level never drops. There is a blood testes barrier protecting developing sperm from material in your blood, similar to like the blood-brain barrier. Protects sperm from the body's leukocytes so that they aren't attacked. Formed from tight junctions between sustentacular cells. Your interstitial spaces surround the seminiferous tubules, and your interstitial cells reside in those spaces. They produce androgens by your LH. Most common one is testosterone. Most of it is released from interstitial cells, small amount from, the adrenal cortex. So you can see the testicle with the seminiferous tubules. Those then connect into the epididymis. There's just a nice histological section. Okay, so hormonal regulation of this of the male system. You have the hypothalamus still releasing GnRH. That's going to stimulate anterior pituitary to secrete FSH and LH. Those two stimulate spermatogenesis and androgen production. LH is going to stimulate the interstitial cells to secrete more testosterone, and the FSH secretes sustentacular cells to secrete androgen binding protein. And this is important because this will bind testosterone to ensure that levels stay high in the testicles to stimulate sperm production. So you need kind of all of this. You need the GnRH to stimulate FSH and LH. LH will then stimulate the interstitial cells to secrete their testosterone, and FSH make sure the sustentacular cells are secreting ADP to bind it and hold it in place. That increased testosterone is going to facilitate spermatogenesis and inhibit GnRH secretion and pituitary sensitivity to GnRH. So it has a negative feedback effect. Again, just making sure that sperm count doesn't get too high, because the more testosterone you have, the more sperm development you have. And the body doesn't want to waste sperm. I mean, even though it can make 100 million a day, it doesn't want to have to make more than it needs to. That could potentially be wasted. Those sustentacular cells will also release inhibin responding to sperm counts, cause inhibition of FSH from the anterior pituitary, and provides additional negative feedback mechanism. Testosterone stimulates libido and development of the secondary characteristics, hair growth, deeper voice from a thickening of the thyroid cartilage, or the laryngeal cartilage rather, and facial hair. Sometimes. Lots of varied secondary sexual characteristics. There is that, you should know the hormonal feedback loops for sure. Okay, spermatogenesis and spermiogenesis. Spermatogenesis is the process of sperm development within the seminiferous tubules. It begins during puberty with those significant levels of FSH and LH from your GnRH being released. You start with your spermatogonia. These are primordial germ cells from which all sperm develop. They are diploid near the base of the seminiferous tubules, surrounded by cytoplasm of sustentacular cells, and they divide by mitosis to new spermatogonium and a primary spermatocyte. So spermatogonia, or plural, spermatogonium is singular. You have a germ cell, again, like I said, is going to divide into another germ cell and a primary spermatocyte that can develop into a sperm. Your primary spermatocyte is a diploid cell that is then going to undergo meiosis. So there is first a mitotic division to produce these, and then you go into your meiotic divisions. Your primary can split into secondary spermatocytes, 2 cells produced by the primary spermatocyte from meiosis 1. These are then haploid cells, so they only have 23 chromosomes, relatively closer to the seminiferous tubule lumen, where they'll be developed. From the secondary spermatocytes, you have spermatids. This is formed when they complete meiosis 2. It is a haploid cell and has a circular appearance. The final stage is spermiogenesis, so the actual formation of a sperm, taking it from a spermatid into a sperm. It becomes a mature spermatozoa, is the official term for like a mature oocyte. It's spermatozoa, secondary oocyte, sorry. Excess cytoplasm is shed and the nucleus elongates. It forms an acrosome cap over that nucleus that has digestive enzymes for penetrating the secondary oocyte. We'll see like I said, the corona radiata. It forms a tail from organized microtubules attached to a midpiece that has mitochondria. Those mitochondria will provide the energy to move the tail. If you remember from lab, though, the sperm are immobile until they actually exit the body. Okay, female versus male gametogenesis. A single viable secondary oocyte is produced in females, whilst 4 sperm are produced in males. The female oocytes are arrested in meiosis prior to birth. The male spermatogonia start spermatogenesis at puberty, so they begin right at puberty. And females only able to produce until menopause, while males are produced throughout the lifetime. So here you can just see you start with your spermatogonium, your germ cell. It will split to make another spermatogonium and a primary spermatocyte. You then have your two meiotic divisions, which then create these 4 haploid cells. And then you have the process, and at that point, you have a spermatid, and then you have spermiogenesis. You have the actual process of it developing its elongated nucleus, the acrosome cap, the midpiece, and the tail. Okay, sexually transmitted infections are a group of infectious diseases transmitted via sexual contact. STIs, because they go unnoticed, usually. Condoms help prevent the spread, but are not 100% effective. It's a leading cause of pelvic inflammatory disease in women, can lead to blockage of tubes and infertility. You have chlamydia is the most frequently reported in the US because of a bacterium. Most people are asymptomatic, which, as we have learned with COVID, just causes even more rapid spread, which is why it's very, very important to know your status. May have symptoms of vaginal discharge, painful urination, back pain, treated with antibiotics. Yeah, that's very common. And again, I don't know why this is solely focused on women. Men will also have discharge from their penis too. Genital herpes caused by the herpes simplex virus, cyclical outbreaks of blister formation in the genital and anal regions. There is no cure for this one. It is an antiviral medications lessen the severity and the length of your outbreaks. Gonorrhea, it's another bacterial infection by sexual contact. It can be spread by this. This is definitely not the only way that it can be spread. It's not only spread rather from mother to baby, but it can be. Some forms are treated with antibiotics, but there is a strain that's circulating throughout the world that is antibiotic resistant. Syphilis, it's a bacterial infection, sexual contact with syphilitic spore, treatment with antibiotics. This one is very important. It will start to eat away at your brain. Okay, that was a nice little detour into STIs. Now back to the male duct system. Your epididymis is the comma-shaped structure that kind of sits around the testicle. It's composed of internal ducts and external coverings. There is a head, a body, and a tail. It has a long duct on the inside of it that stores sperm until they're fully mature and motile. That's really the important part you need to know. The ductus deferens, your vas deferens, connects the epididymis to the urethra, essentially. So sperm enter this after leaving that. It's located within the spermatic cord, and it runs superior up into the posterior surface of the bladder. I'm sorry, superior lateral, so kind of on the side into the back a little bit. Surface of the bladder. It enlarges and forms the ampulla, unites with the seminal vesicles to form the ejaculatory duct that inserts into the prostate gland. Again, if you remember from lab, we went over a lot of this, you saw a lot of this. There's an inner mucosa, a muscularis layer, necessary for moving sperm. Remember, sperm are not motile until ejaculation, so it's important to have that smooth muscle to provide contractions and movement. The ejaculatory duct, it's between 1 and 2 centimeters long. It conducts sperm and a component of seminal fluid towards the urethra, and again, it opens into the prostatic urethra. That's where it's inserting into. The urethra transports semen from the ejaculatory ducts to the outside of the body, as well as urine. It has 3 components: prostatic, membranous, and spongy. They're named for their specific locations. Yeah, here is kind of a just vertical view of it. You have sperm produced in the testicles, stored and matured in the epididymis, released through the vas deferens, where they're inserted. So this is a posterior view. You have the seminal vesicles and the ampulla of the vas deferens forming into the ejaculatory duct on the prostate. You have your prostatic urethra, your membranous urethra, and then your spongy urethra that runs the length of the penis. All right, so seminal fluid. This is an alkaline secretion needed to neutralize the vagina's acidity. It gives nutrients to sperm as well to travel through the female reproductive tract. We'll see that sperm can live longer than just the 24 hours that an unfertilized oocyte can. Because they have these nutrients in the seminal fluid. It's produced by different accessory glands of the male tract: seminal vesicles, prostate, and bulbourethral glands, also called Cowper's glands. The seminal vesicles, posterior of the bladder, it secretes this alkaline fluid that has both fructose, a sugar, and prostaglandins. the fructose nourishes the sperm. The prostaglandins promote the widening of the external os of the cervix, so they help it to dilate. The prostate gland is a walnut-shaped encapsulated organ immediately inferior to the bladder. It produces mucin. There are tubuloalveolar glands that contribute to seminal fluid, secreting this rich in citric acid, seminal plasm, and prostate-specific antigens. So the citric acid is nutrients for sperm health. The seminal plasmin is antibiotics, and PSA is an enzyme to liquefy semen following ejaculation. We'll see that semen is seminal fluid and sperm mixed together. So you have your seminal vesicles, your prostate gland, and then your bulbar urethral glands. These project into the base of the penis, entering the spongy urethra, produce a clear viscous mucin that forms mucus to help coat and lubricate the urethra during intercourse. Semen, again, is formed from seminal fluid and sperm. It is called ejaculate once it is released during intercourse. That contains 200 to 500 million spermatozoa. The transit time from the seminiferous tubules to ejaculate is about 2 weeks. So again, from like formation to actually being able to be released, it takes about 2 weeks. Benign prostatic hyperplasia is a non-cancerous enlargement of the prostate gland, very common in older men due to hormonal changes. Large nodules form within the prostate, compressing on the urethra. Frequent urinating at night can be painful as well. Can be treated with drugs, may require surgical removal of prostatic enlargement. After you're 50, get checked every year. Oh, there it is. Prostate cancer. Sorry, I should not laugh when I say that. One of the most common malignancies in men over 50. Forms of hard, solid nodules within the prostate. Asymptomatic early on. Later you get urinary symptoms. Typically, one of the common symptoms is that when you are urinating, for men, typically it is that you can start urinating in a nice, strong, steady stream, and then as you finish, it will start to drip. As the prostate starts to enlarge because it's compressing on the urethra, typically what happens is it starts to begins as a drip and then becomes a steady stream. That's usually when it's time to start definitely getting your prostate checked. Screening with a digital rectal exam. Treatment is radiation therapy or surgical removal. Okay, genetic sex versus phenotypic sex. Genetic sex is based off of your genotype rather. So it's your sex chromosomes that are inherited, determined at fertilization. So you can have an XX, which is a genetic female, or an XY, which is a genetic male. The phenotypic sex refers to the appearance of an individual's internal and external genitalia. So ovaries and female external genitalia, you are phenotypically female. Testes and male external genitalia, you are phenotypically male. This is usually apparent by the seventh week of development. It's important to note these terms. Male and female are biological sex determinations based off genetic sex. Woman and man, boy and girl, those are phenotypic designations. A lot of medical texts still kind of have not caught up to this distinction yet, but it is one that a lot of social psychologists and a lot of more modern teachers are going to be talking to you about. So having a female and male and making that distinction is important because as we are highlighting more transgender rights and looking to try and make medicine more inclusive for all people, it's important to understand how using pronouns and how using gender terms like man, woman, boy, girl versus biological terms like male and female do impact people and how they do hear these words. And as a healthcare professional, especially if personally, if you are not concerned about this, then that is, your personal belief. But if you are going to be a healthcare professional, the way that you talk to people and the way you initiate conversations is really going to determine the quality of care that you can provide. If someone does not feel, even just from the way that you're speaking about gendered versus non-gendered terms, that is going to impact what they say to you, how honest they are to you about what they're going through, what their symptoms are. And if they don't feel comfortable, they're not going to disclose and they're not going to be honest, and you won't be effective at your job. So again, if you want to be good at your job, this is just one of those layers that you can learn. And if you can learn all the other stuff in this course, this is not a hard thing to learn. So just to put that out there. The other thing, based on the appearance of an individual's internal and external genitalia, it does not happen as much anymore. I'm not going to say that it doesn't happen, but intersex surgeries are a common thing to hear about at birth. If a child is born and it's slightly unclear whether the external genitalia is going to be male or female, because we don't do genetic testing on every child to determine whether or not it actually genotypically is male or female, if it is genotypically XX or XY. Most determinations of sex are based simply on external appearance. And sometimes that is not always as clear as you may think it is. And in the past, what doctors usually would do was they would make the decision, no consult with the parents or anything. They would look at it, say, "Oh, yep, this looks more like a boy to me, so we're gonna treat it that way, whether with surgery or hormones, whatever it may be." And there you go, you have a boy. Congratulations. That has become more controversial, and there's a lot of debate that you can look into on that. And I will go ahead and say, if you have questions about gender versus sex and identity, transgender questions around transgender populations, I do not speak for transgender people. I am not representative of the population at all, but I can help you answer questions, and I can point you to resources if you would like them. I would love to have that conversation with you. Okay. So your sex determining region. So what actually does then cause for this differentiation? Because there is a difference in between a male development and a female development, both structurally, anatomically, and physiologically. So on the Y chromosome, there is something called a sex determining gene. It is located larger within the testes determining factor region on the Y chromosome. In the genetic male with a Y chromosome, the SRY gene is expressed, produces proteins to stimulate androgens, and those initiate male phenotypic development. If the Y chromosome is absent or lacks or has an abnormal SRY gene, then phenotypic sex results will develop. So you can see you can have an XY female depending on what the just the expression of the specific gene. Developmentally, you can then move to puberty. This is a period in adolescence where reproductive organs becoming fully functional. External sex characteristics become more prominent, your secondary sex characteristics. Time affected by genetics, environmental factors, and health of the individual. This is one of those kind of strange things. I think, at least for me, what seems like a common example is gymnasts. So female gymnasts, because they tend to go under such harsh training regimens and their diets are so strictly controlled, they typically do not enter puberty until later. It can be 14, 15, 16 before they're actually having their first period, which then, of course, does affect all future development. Not sure how much, but it does affect some. I won't be that reckless to say that it's going to just throw it off completely. All right, so initiation of puberty begins when the hypothalamus starts to secrete GnRH. Again, that's going to cause the release of FSH and LH. Up until then, the levels stay very low. They stimulate significant levels of sex hormones to start the process of gamete and sexual maturation. The early signs of development, again, you can talk about breast development, pubic and axillary hair, menarche 2 years after the first signs of puberty. You start to have testicle and penis growth, rapid growth of laryngeal structures, causing voice changes. I'm sure we all remember a range of these. Usually, females are going to be 2 years prior to males, African Americans before Caucasians. Onset has dropped with better nutrition and healthcare. About 8 to 12 for girls, 9 to 14 for boys. You can have precocious puberty, which is signs of puberty developing much earlier than normal, maybe without a known cause or due to brain injury or a pituitary or gonad tumor that are causing increased secretions of those hormones. Right, so now kind of continuing towards the end of it, you have menopause. But perimenopause is the time near it, marked by irregular or skipped periods. Then full menopause is when a woman starts monthly menstrual cycle for a whole year, usually between 45 and 55, atrophy of reproductive organs and breast with reduced hormones, decrease in vaginal wall thickness, and the uterus starts to shrink. You can have hot flashes with periodic elevations in body temperature, thinning scalp and hair, increased facial hair, risk of osteoporosis and heart disease. The symptoms can sometimes be treated with hormone replacement therapy. which many physicians have stopped prescribing. Men do experience something nowhere near similar. Let's be very clear. It's the male climacteric. It's decreased testosterone levels in their 50s due to decreased interstitial cells. Decline is more gradual. Most men have very few symptoms. Some with mood swings, decreased sex drive, and hot flashes. The prostate may start to enlarge. That's a common experience, though, for all men. It can interfere with sexual and urinary functions. Another thing, erectile dysfunction and impotence. Erectile dysfunction is the inability to achieve one. Impotence is not being able to get one at all. And associated with aging and other risk factors. All right.